I'm Alexis van den Bon from Kyoto University. I want to present about the basics of spatial data analysis using R. And then also I want to introduce several spatial transcriptomics databases. And so the goal of this webinar is, I hope I can explain the basics of spatial transcriptomics data analysis. And I want to introduce those databases, including the database that we have been developing by ourselves called DeepSpaceDB. And so the target audience, I think it's similar to what Dr. Sakai just introduced, but of course, it's people who are interested in spatial transcriptomics data analysis, or people who want to know what kind of databases exist or how to use them. And here I should say, so I will show some R code, but I will not explain the details about R. So maybe it's useful if you have some basic understanding of R programming already a little bit. And then I also want to briefly introduce myself. So I'm now in Kyoto University, but I, of course, was originally born not in Japan, but in Belgium, close to a city called Bruges. And this is a picture from Bruges. It's a famous tourist spot. It's like a medieval city. It's very beautiful. But so I already came to Japan about 20 years ago. And first I joined a PhD course in the lab of Professor Kenta Nakai. And after that, I joined Osaka University. And then several years ago, I moved to Kyoto University. And now I'm an associate professor here. And of course, I'm doing research in bioinformatics. And then, so this is the content for today. So briefly, I want to show a simple example about R and how to analyze a Visium sample. And then I want to introduce several databases and more deeply should show about our own database. So I think some of you will be familiar with the single cell data analysis. And as Dr. Sakai just said, some of the analysis tools that we use for spatial data analysis were originally made for single cell data analysis. And one of them is maybe the most famous package for this is called a Sura. And so I will use this as a basis for my presentation here. So if you go to their website, maybe you know there are several tutorials there. If you move down, so you will see 3 tutorials about spatial data analysis. One is for image-based spatial platforms. One is for Visium HD. But one of them is sequencing-based spatial transcriptomics analysis, including the 10x Visium platform. So I want to use that one as my example. And you can also find this if you go to vignettes, and then the same tutorials are also there. And so I will use that as a basis, but also so I put the code that I will show on GitHub. You should be able to access it here. And I limited the code to just the really essential commands. So I removed things that are not really necessary. I hope it's easy to understand. So, and this is a typical workflow that we might use. So first, of course, I have to read in some data set of interest into my R session. And then I should do a quality check. Or maybe some parts of the sample are not so good. I want to check it. And then normalization. And then I guess comes the more important part here. That's exploring the data, exploratory data analysis. And this is especially important for high dimensional data. So here, in this, for example, 10x Visium sample, we have data for maybe almost 20,000 genes. And every gene is, in a sense, one dimension. So it's really hard for humans to look at 20,000 dimensions. So to explore this, maybe we want to reduce the dimensionality, or we want to group the spots or the cells by similarity. into maybe just 5 clusters. And then it becomes a lot more easy for humans to look at just 5 clusters and check if the data makes sense. So that exploratory data analysis is really important to check, does the data look okay? What are the main features of the data? And while I'm doing it, I'm not running a statistical test yet, so I'm not testing hypotheses, but I might try to generate hypotheses. Maybe I want to check this or that later. And then also, I should say there's no single way that's correct in doing this. So there might be several ways for doing this exploration. So in that sense, it's rather difficult, I guess. There's no correct answer. Then, so I will use a 10x genomics Visium example in my lecture today. So I should briefly explain about it, although maybe some of you will be familiar with it. Of course, it's made by a company called 10x Genomics, and maybe they have a slide that looks like this. And here you can put your tissue slice. Here they use an example of a mouse brain. And this region, so it's covered by small spots. And here they zoom in on those spots. So they follow a hexagonal pattern. And on each spot, there are DNA molecules that have a unique sequence. We call it a barcode. Every spot has a unique barcode. So it's like their address. And then we let the RNA from the tissue bind to those DNA barcodes. And after several steps, we sequence the RNA part and the barcode together. And by looking at the RNA part, of course, we know which gene was transcribed. And by looking at the barcode, we know which spot it came from. It's like the address of the spot. So at the end, we will get data that looks like this. For every spot, we know how many reads did we see for every gene. And of course, also, we know where is each spot. We know the X and the Y the Y coordinates of each spot. So using these two data types, we can reconstruct for every gene the spatial pattern, so the expression pattern within the tissue. And here I should also point out, so the diameter of these spots is 55 micrometer. So it's relatively large, right? It's bigger than a cell. And maybe 10 cells will cover each spot. So each spot shows the average of 10 cells, something like that. It's not single cell data. So now I want to first read in some sample, or I load some sample that I'm interested in, and then check the quality. And here I'm showing the R code that we might use, so which you can see on GitHub also. And so, of course, I want to use a Seurat package, so I have to load it first. And then reading in the data can be quite difficult. So here I try to keep it simple. There's another package called Seurat data, and it includes several data sets that you can relatively easily load into your R session. So I have installed Seurat data, and here I load it, and then I install the data set that I'm interested in, STXospatial brain. And then from that brain data set, I want to load the sample called anterior one. So it's the frontal part of a mouse brain. And I put this into the brain object. Okay, so here I'm reading in this data using the Seurat data package, which makes it relatively easy. Okay, and then I want to check the quality of that data. And if you are familiar with single-cell data analysis, you will notice this is very similar to single-cell data analysis. I make a violin plot of the number of reads per spot and count, and that's plot 1, so this one. And then the same thing for the number of detected genes, so N feature. This is the second plot, this one. And also here I'm making a scatter plot where the x-axis is the counts and the y-axis is the number of detected genes, which is that third plot. So I think if you have performed single-cell data analysis before, these will look very familiar to you because we might do the same thing for single-cell data analysis. And we can also see that there is some variation between the spots. So here, every point here is one spot. And you can see there's quite some variation between the spots, right? So some have a very high number of reads, and some have a very low number of reads. Some have many detected genes, and some have a very low number of detected genes. Of course, there's a clear relationship between those two, with a certain plateau is reached here. So in single cell data, so some things are different from single cell data analysis. In single cell data analysis, I might say I will remove these spots with a low number of detected genes because maybe they are dead cells. And also, maybe I will remove the cells with high detected number of genes because they might be doublets. I cannot explain in detail now, but we get the micro droplets, and they might contain not one gene, not one cell, but two cells. So we call it a doublet. And in single cell data analysis, we want to remove those doublets. But here, so this is not single cell data analysis, it's spatial data analysis. There is no concept of a doublet. So I don't want to remove those spots with a high number of detected genes. So the interpretation of these quality measures is a bit more difficult than in a single cell data analysis. And I can see that also if I plot the same quality measures, so the number of reads and the number of detected genes, in a spatial plot, a spatial feature plot, this generates these two plots. So it shows the same quality measures within that tissue slice. And now I can see that this clearly reflects some structure of that brain tissue. There's a clear, these spots have a lower value, and there's a lower range here, and these are high, and these are high. So it reflects the biology of that tissue, and not necessarily a technical problem. So the difference in the reads might be caused, for example, because maybe the density of cells is relatively low here compared to here, or there are different cell types. Or also so several slides ago here, I said I have to make this tissue permeable so the RNA can bind to the DNA. And maybe some parts of the tissues are more easy to make permeable than other parts. And this can also lead to these differences in the number of reads or the difference in the number of detected genes. So we have to be are a bit careful in how we interpret these quality measures. And here I want to show some more examples about that. So I took some examples from our database, and these are 3 different mouse brain slices produced by different studies. And I'm showing the number of detected genes. And you can see there's a consistent pattern here. So in different studies, they find the same variation in the number of detected genes, suggesting that this is not a technical problem. This is a biological feature of that tissue. And here also, so I'm showing a hepatoblastoma sample with also the image. I guess here is a cancer tissue. And clearly there's more detected genes around that hepatoblastoma. So I don't think this is a technical problem. This shows the biological properties of this tissue slice. So I should not exclude these spots because they have a high value. I should not exclude these spots because they have a low value. So on the other hand, so in some samples, we can see a clear technical problem. So here I'm showing one example. So this is a liver sample with metastasis from breast cancer. And this is the image of that tissue. It looks perfectly fine, I think. And here this too looks okay. But at the border, there's a straight boundary here, where suddenly the number of detected genes is very low. I don't think this reflects the biology of the tissue. this probably reflects a technical problem. So I should remove those kind of spots before I continue my analysis. Okay, so I'm going back to that tutorial example now. So in that tutorial sample, there is no problem, but I should normalize the number of, or I should normalize the data before I can continue my analysis, because there's a difference between the total number of sequences in every spot. And I cannot explain in detail, but there are several ways that you might normalize your data. Maybe this one is easy to imagine, so option 2. I correct for the library size, so the number of reads per spot, and that's option 2 here, which is commented out, so I'm not using this one. The people who made Seurat, they insist that this is a better way for normalization, although it's a little bit more complicated, so that's what I'm doing here. And after that, so I want to look at the exploration of the sample. So it includes dimensionality reduction and clustering. So I want to go from a high dimensional, like complex data to a summary that is easy for humans to understand. So here we run PCA, and then we cluster, so similar to what Sakai-san showed. So we detect the neighboring spots, we cluster them, and then I make a plot of it. So I do spatial dim plot. This makes plot number 1, so that's this one. And so the different colors here show the different clusters. And I think you can see there's a very clear pattern here. So this is cluster 7, and cluster 4, and cluster 6, and cluster 3, and cluster 2. And here there are 2 clusters in the olfactory bulb of this brain sample. If I compare those clusters to what we know, so I took this from the Allen Brain Atlas. So this sample covers roughly this region. Actually, it's very similar to what we got. So even though we just clustered the spots by similarity of their gene expression pattern, we get something that's really close to the known structures within a brain sample. And then, in addition to that, so I can run UMAP and try to show the data in just a 2D plot. And that's what I'm doing here. So this is making plot number 2. And here again, so each dot, each point is one of the spots. And now I can see there's also some structures, right? Or maybe there's one group here and another group here, representing roughly these parts and these parts. And actually here I can see, so here I have cluster 2, 3, 6, 4, and 7. Here too, I can see 2, 3, 6, 4, and 7. They are somewhat connected in this way, showing how they are comparatively similar to each other, right? So using those kind of steps, I can get some idea of what is inside this data. I will skip this slide in the interest of time. And then, so maybe the next step that I want to do is I want to predict genes that have a clear difference in expression between the clusters. And so one way of doing that is use the find markers function, so which is again the same as we use in single cell data analysis. And here we are saying, for example, I want to compare between clusters between 2 clusters, so cluster 5 versus cluster 6. And then we predict the markers, and here I'm showing the top 3 scoring genes. And here is cluster 5, here is cluster 6. I can see that in each case, so there's a clear difference between their expression pattern in 5 and 6. So, of course, I can use this to predict genes with differences in expression. But, so I have to stress that this is not using the spatial data at all, actually. And so far, in none of those steps, we use the spatial coordinates of the spots. Another way for predicting interesting genes is to look at their spatial pattern. So, and here, this is the first step where we are actually using the spatial information. So there are several ways for predicting these spatially variable genes. They are genes with a clear spatial pattern. One is called Moran's eye. So it's looking at autocorrelation. It looks like two spots that are next to each other. Do they have a similar value? And there's other methods like SparkX or BinSpect or single cell haystack, which is a method that we developed by ourselves. And here in this example, so I'm using Moran's eye, this one. I should say it's relatively slow, but we predict the spatially variable features, the genes, and then again we plot the top 6 ones, which are these 6 genes. And of course, we can see that they clearly have a spatial pattern. So this one has a high expression here and not here. And here is similar, so here and not here. This one very high here, but not here. So of course, it works. And now finally, actually, we are using the spatial component of this data. So here, I will stop the introduction of R. Of course, next, similar to what Sakai-san showed, maybe I want to predict the cell, what are the cell types or things like that. So, but I will not introduce it today. And then there are other platforms, of course, which Surat can handle. And so I introduced Visium, but there's also a tutorial for SlideSig. And then there are also tutorials for the image-based platforms, like including Xenium, for example. But actually, the workflow is very similar for each of them. So as a conclusion for the tutorial by Surat, of course, it's really nice. I think it's a very useful package for analyzing single-cell data and also spatial data. And the workflow is relatively similar. But so as I try to show, the spatial component is actually not used so fully. And of course, we can do some interactive analysis, but it's rather limited. So, and I think maybe what we actually want to do is, for example, I want to click on a spot in the tissue and I want to see which genes change very drastically around that spot, for example. Or I want to draw a line using my mouse and I want to see, what changes along that line, for example. Or I want to select some regions within a tissue slide and I want to see which genes have a different expression. So I want to select them rather freely. But those kind of interactive analysis are really difficult, even now using Surat and also using other packages, I think. So next, I want to move to the second topic, which is about spatial transcriptomics databases. And so I should say already several databases have been developed and made public. So here I'm showing several of them, including our own database. And so the URL, so you can check it later, of course. And the publication with a link to the paper. And the ones indicated in a star, I included in the slides. But I'm afraid my talk will become very long if I introduce each of them. So I will go through most of them rather quickly, I'm afraid. So the first one is SpatialDB. So it was maybe the first database that was developed. And it covers several studies and technologies. But as far as I can see, so it has not been updated since the publication. And as a result of that, I guess some of the tools are no longer responding as far as I can see. Then we have SLDB. And this one, so it also covers many samples and different technologies. Sometimes it's a little bit slow, but so it includes transcriptomics data. This is from their paper, but also proteomics data. So that's really interesting. And you can click here and see an overview of the technologies that they cover. And you can also browse or use filters to select the species of interest or the tissue of interest and select a sample. And you can see a summary of the sample, for example. And you can give as input a gene of interest, and then it will show the expression pattern of that gene within the sample. However, so as far as I understand, So if you hover with your mouse cursor above these spots, it shows the value. It's always an integer, which suggests to me that this data has not been normalized, actually. And also the image is not shown or not included. So I think unfortunately, it's not so useful in practice. That's my impression. And there's a few other things that you can do using this database, including seeing a clustering result, for example. Then we have STOmicsDB. This one is, as far as I can see, the biggest one. So it covers data from more than 350 studies and different technologies, and it includes 15,000 samples, so it's really high. Although I should say, so they give a quality score to every sample. So some have 1 star, some have 2, up to 5. And here's actually many of them have a low number or a low quality. And 2 stars means so it does not have the spatial coordinates and it does not have the image. And 1 means not processed. So actually, I think these samples are not so useful in the end. So you can filter for your technology of interest. So for example, I select here mouse and then brain tissue and stereosig, so which is one of the platforms. And then you can pick one of the samples that you are interested in. And you can see, for example, the clustering result. This is a mouse brain sample. And for some of the samples, actually, they added annotation data. So it's really nice, I think. You can also fill in a gene of interest. It will show the expression pattern of that gene within the tissue. And even so, you can use this lasso tool to select a region of interest, and it will show like the average expression within the selected region. So that's also nice, I think. There's also links to the original data, and you can download the data and maybe analyze it by yourself on your PC. Well, that was a stereosig example. Well, I can also show a 10x Visium example. So this is a human and breast and 10x Visium. And then select one of the samples. And here, so I can similarly see the clustering result, for example. And I can similarly input a gene of interest and see its expression pattern. And there are some things that are different depending on which technology the sample is based on. So this was not available for the previous sample that I showed. So this is maybe only present for the Visium platform. So I can see the quality indicators similar to the violin plots that I made in the first part of this presentation. Or I can see the cluster markers. So they predict the genes with a different expression between the clusters. And if you click them, so you can easily visualize them. And there's something called hotspot analysis. I guess it's the clustering of genes by similarity. You can see it in a heat map, and you can download the result. And we can check that, for example, this gene is in module 3. And module 3 looks like this. And we can confirm that this gene, the expression pattern fits with module 3, for example. And they also have spatial markers, so predicted using this spatial DE method. So these are spatially variable genes. And again, if you click here, you can easily visualize those marker genes. Okay, this is the last one, so it's called SOAR, I guess. This one also, it has several different platforms and many samples. And you can click again, you can show the image, you can see the clustering results, you can visualize a gene. So the features are rather similar for every database. And actually, this one also includes a few Xenium samples, maybe eight Xenium samples. So you can plot a gene again, like this one, and you can see maybe spatial domain predictions. So, but for example, you cannot zoom in, or you cannot do some interactive analysis, actually. So, as a summary for this part, I think the good points about those databases are they include many different platforms, and they have many samples. but I feel that they also have some problems. So the analysis functions are rather simple. I can plot a gene, or I can see the clustering result. And I feel the interface is not so intuitive and maybe not so pleasant to work with. And also it includes some low quality samples, actually. So I think those are some weak points of those existing databases. And that's why, so of course, we try to develop our own database, so DeepSpaceDB, and we hope to address those weak points and make an interface that is more interactive and maybe more easy to use. So, and now our database includes two different platforms. So one is a Visium and one is a Xenium. So I want to introduce both of those interfaces in some more detail. Now, this is what we did. So first for Visium. So we collected public data from several sources, such as GEO or the 10x genomics database and other sources. And of course, it includes the image and the transcriptome data, and then annotation data. I will introduce this in a moment. And then we processed every sample using a fixed pipeline, and then added it to our database. So, and I said, so we did the annotation. So we really try to do this manually as far as possible. So for example, if the sample was collected from GEO, we will go to the series that produced that sample, and we will check, so what's the species, of course, and then it says dystrophic muscle, so I guess it's a muscle tissue. And it says, so severely dystrophic D2-MDX mouse model, so we will write that down also. And then we link also the paper connected to this series. And then maybe we will check, so how many samples are there, and for every sample, we will open the sample page and check, so when was it published? And okay, this is the genotype MDX, and the tissue is gastrocnemius. Okay, and then we collect all this annotation data, and that's what's shown in our database. And of course, as you know, so somewhat different people might describe the tissue using different words, so it might become very inconsistent. And the same thing for the condition, actually. So we try to make this consistent by using existing ontologies. There's something called the Uberon anatomy ontology. So we check, is gastrocnemius, is it in this ontology? Yes, it's in it. So we link this sample to this term in that ontology. And the same thing for muscular dystrophy. Is it in this human disease ontology? Yes. So we link this one to that term in that ontology. So I think it's nice, but of course it's very time consuming. And of course we are not specialists. I don't know about this muscle tissue at all. So in the future, we hope to try to use the LLMs to partly automate this step, actually. It's one of the things we want to do in the future. And this is the current number of samples, so the Visium samples in human and mouse. So for every tissue here, I show how many samples we have. So in total, it's more than 1,300 human samples. And the color is showing the condition. So, and you will see many of them are red, and they are cancer-related samples. So especially for human, many samples are related to cancer. And for mouse, there's more black color here. So it means healthy or control samples. And especially brain is very popular for mouse. But our database covers many different tissues and many different conditions. And next, I want to, so these samples were collected by many different groups all over the planet. So of course, we wonder, so can I really compare between them? So maybe there's a huge difference in in the conditions that were generated, that under which those samples were generated. So there might be batch effects, right? So we try to check. So for example, if I take all the samples, so every sample, I turn it into a pseudo-bulk sample. So I average the values of all the genes across the spots. And I try to give this to, for example, t-SNE and make a typical t-SNE plot. So what would it look like? So each spot here is one human sample. And there must be 1,300 spots here. And they are colored by the tissue of origin. And so, of course, we can see that samples from the same tissue are close to each other. So it means, in general, their gene expression looks similar. So I think it makes sense. So here is all the brain samples. Here are the skin and mucosa. Here is the heart or kidney and liver. So that makes sense, I think. Yeah, if this was completely randomly distributed, it would be a problem, I feel. But it makes sense. But here, so many of them are maybe looks more shuffled. But actually, these are especially rich in the cancer-related samples. So I guess the cancer makes this pattern not so clear. So on the other hand, for the mouse samples, it looks really beautiful, I think. So liver samples are all together, small intestine and brain and large intestine and thymus, lymph node, spleen. So I think it makes sense. I think this data is relatively clean, is my feeling. So we collected those samples, and then so we prepared the database, of course. So this is the top page of the database, and it has 6 tabs. So one is database, where you can search for a sample of interest, and then search, I will introduce later, so you can search with a gene of interest, the whole database. Then upload, you can upload your own Visium sample and analyze it on our website. And then there's a tutorial about and contact. So I want to introduce several of those things in some detail, but first I want to show a video demonstration. So this is the top page, and I go to database, and there's a table with all the samples. And if you click here, so it will show a preview, and I can add a filter. So I'm saying human and breast. This shows all human breast samples. So they are all breast cancer samples. And the ones that have a yellow star, the image has been annotated by a pathologist, a human specialist. So maybe let's select this sample. It brings you to the sample page, which has many different kinds of information. And first is the metadata, so the annotation. So this is human breast cancer from this study. And I will skip some things. This is the quality indicator. So this is number of detected genes. And this is the annotation by the specialist. So there's a tumor tissue here, for example. And it's clustering. And these are the spatially variable genes. These have been pre-calculated, so I can just click them and immediately check the pattern here. This is high here, for example. And we did the same thing for pathways. So we try to estimate the activity of every pathway, and then you can easily visualize them here. And then maybe the most interesting tool is the Tissue Explorer. So here I can select with the mouse, so freely a region of interest. I say, okay, this is set 1, and add it to the set. And this shows my current selection. And then, so for comparison, I selected a second region, and I added to set 2. So these are the 2 sets I selected so far. Then if I click compare gene expression, it will calculate the average for every gene in those 2 regions. And then it will show it below in the result. So it shows it in a table and in a plot. So every gene here, every spot here is 1 gene. And so the set 1, the first region I selected was the cancer tissue. So I can see one of the genes that's high here is DCAF7. So I can go back to the genes tab, and I can search, so DCAF, and here is DCAF7. So if I click it, I can confirm that this gene has a high expression here, the first region, and not in the second region. So of course it works. I think it's really useful for exploring the tissues. And then at the bottom, there are several files that you can download and analyze on your own PC. So that was a short video. And now I want to go through some of those parts more slowly. So first, this is the table I showed. So you can see all the samples, you can see a preview, and you can use filters. And you can also search for a keyword, like Alzheimer's disease, for example. And then you can add the filters like human or breasts or cancer. So this will give you the breast cancer samples. And then you can scroll through the samples, of course. The yellow stars means there is an annotation by an expert. And then if you click sample page, so it will take you to the sample page. So, and as I said, there are many kinds of information here. So first is the metadata, right? And then below that, so there's the location of the sample in the database. And this is showing this plot, which is the same plot that I showed before. So this is based on the pseudo-bulk data. And here, so these plots are interactive. So you can click and select a region, and it will zoom into that region. And so this red one is the sample that I currently selected, so the breast cancer sample. And the samples that are close to it, so it means they have a similar gene expression in general. So if I click one of them, they will appear here next to this plot. So I think you can use it for finding similar samples. Then there's the quality measure. So I can show the detected genes, or I can show the read count. I can show the percentage of mitochondrial reads. And so here, of course, I can see some regions are high and some regions are low. And here, especially at the border, some spots have very low numbers of detected genes I can see. So it might be a problem. Then those are those image annotations by an expert. So this was done by Dr. Horimoto Yoshiya. He's from the Tokyo Medical University. And so he kindly annotated almost 70 breast cancer samples. But I should say, so these H&E images are often of a relatively low quality. So the resolution is not so good. So he annotated them as far as possible. But there's a limit to what he can do because the resolution is rather low. And in addition to that, so if you click here, you can see the annotations made using a large language model. So this is what we did. For every image, we cut it into a grid like this. And then we give each part to GPT and we say, so please tell us what you see here. And we collect those annotations and put them in the database. So this is what's shown here. But of course, you have to be careful with those annotations because I don't know if it's really reliable. Maybe this will improve in the future. But anyway, so we are trying to help the user understand what is inside each tissue. That's the goal here. And actually, we can see, for example, those image annotations, they fit very well with what we saw for the number of detected genes or the mitochondrial reads or the read count. So clearly, this is reflecting some of the structures within the data. Then, so we have the clustering result. So we cluster the spots of each sample and then show the result in the tissue, but also in the 2D embedding. And here too, so I can see that these clusters fit somewhat with the annotations by Dr. Horimoto. So in addition to that, we also took all the spots of all the samples and clustered them. Now these are millions of spots. And then I tried to give an annotation to each cluster. So for example, we checked for every cluster, how much does it overlap with every tissue? Or how much does it overlap with each condition? Or which marker genes are expressed in every cluster? And then based on these, we try to give a global annotation to each of these clusters. Now, maybe they are neurons or they are tumor and so on. And so in the database, if you select this part, you can see those global clusters, the database-wide clusters. So I'm showing them here. And here, so I'm showing only the ones that are related to tumor, which we annotated as tumor. And you can see that, again, this fits relatively well with the annotations by the expert. So I think those clusters can also help the user understand the content of each sample. And we also added spatial domains, so using the predictions of two methods called BAS and BaseSpace. But so I think maybe Dr. Sakai will talk about this in the next part of this webinar. So I think there are many features, but then, so the next one is spatially variable genes. So we predicted spatially variable genes using three methods: a single cell haystack, so our own method, and SparkX and BinSpec. So the user can select which method you want to use, and the results have been pre-calculated, of course. So you can easily click and then show those spatially variable genes very quickly. And here, maybe I very briefly want to explain how single-cell haystack works. So if you are interested, please look at these two publications. So it tries to predict the spatially variable genes by looking at the distribution of cells and their expression of the genes. So let's use this one as an example. So this is a mouse brain tissue. And maybe this is a clear spatially variable gene. It has a high expression here and not here, for example. And on the other hand, this is not a spatially variable gene. It's expressed rather randomly everywhere. So in our method, first, we detect where are the genes, where are the cells in this space. And here, so this is just an image. I made this by hand, but in my imagination, it looks like this. And then for every gene, we also estimate, so where is the RNA of this gene? So, and in this case, the RNA would be really concentrated here and not so much here. While for this gene, it's relatively present everywhere. And then we compare these distributions to this distribution. And this one is completely different, and this one is rather similar. The genes that are different, those are spatially variable genes. So that's the concept of our method. But if you want to know the details, please check these two publications. Okay, so we also did the same thing for pathways. So for every pathway, we take the genes that are known to play a role in those pathways, and we predict their activities, and then find which pathways have a clear spatial pattern. And again, by clicking, you can easily see them. So for example, ATP metabolism is really active in that invasive tumor tissue, I think. Then we also predict the cell types. So using two methods, one is called RCTD, one is called Spotlight. You can click here and show the predictions, although it's a bit hard to see. I want to explain how it works, but I don't have enough time, I think, so I will skip it. Please check this publication if you are interested. But basically, it compares each sample to a reference sample and tries to see for each part how well does it fit with the reference. And actually, it's not so easy to pick a suitable reference for every sample. So it's one of the difficult points in this analysis, I think. And then, so we have that Tissue Explorer tool. So I already showed this, so you can select a region of interest or several regions of interest. Here it's shown the current selections. And then after you made several selections, you can click here and compare between the two or more regions. I think you can select up to maybe 10 regions. And then it will show the result here. So it shows a table with the averages and a statistical test p-value. We just use a t-test. And it shows a scatter plot and also a volcano plot. And then if you hover above each point, you can see which gene it is. And so here I can confirm that those genes have a clear difference in expression between the regions that I selected. Then finally, on the sample page, there's also cell communication data, which we predicted. And also, so you can download several files and analyze them by yourself on your own PC. Okay, that was the sample page. But we also have a search page and an upload page. So on the search page, you can fill in a gene of interest. Now, maybe you're interested in this gene. And then you can search the whole database. So it searches through all the samples, and it returns the samples by how strong is the pattern of that gene in all the samples. And it shows it as a figure and a table. So this is the order of the strength of the spatial sample, the spatial pattern. And you can see that many of the samples with a strong pattern, they are liver samples. Here too, I can see the top scoring samples. They are all liver samples. And then if you click here, it will show the expression pattern of that gene within those samples. And we can confirm that it has a very clear pattern here. I think this could be useful for getting hints about the function of a gene. So I know this gene is important in the liver. So I could confirm that by seeing that these are all liver samples. And maybe it can also help you to find samples that have a very clear pattern. Then finally, there's also the upload tool. So you can upload your own Visium sample and then process it. And after some time, you can see it in the same interface as I showed for the public data. But I should say, so if you upload your sample, it does not mean it will become public to everyone. And also, it does not get added to the database. So please don't worry about that. You can remove it anytime, actually. And also, if you don't remove it after 1 week, it will be removed automatically. Okay, that's about the Visium interface. But I also want to quickly introduce the Xenium interface. So as you know, Xenium has a far better resolution than Visium. Here I'm showing the same gene in two similar samples. One is Visium and one is Xenium. The pattern is very clear in Xenium, but not so clear in Visium, and the same thing here. So we are collecting many public samples from sources, and so far we processed 628 Xenium samples successfully, and we are again annotating the samples and adding it to the database. But here, so one problem is the size of the data, actually. Here I'm showing the distribution of the number of cells per sample. And on average, it's 120,000 cells. And the biggest sample has about 1,700,000 cells. So it's quite big. And for comparison, so the Visium spots is just around here. So it's maybe 1, 2, almost 3 orders of magnitude bigger sometimes. And so in the database, we have to be able to quickly access the data and show it to the user. So we have to do some preparations to do that quickly. And so how do we do it? We use something called a ZAR store. It's a format that's useful for accessing big data very quickly. So each spatial sample is like a 3D data set. It has the X and the Y coordinates, and then the genes are a third dimension. And in a ZAR store, so you can chunk or cut this data into small parts and save them separately. And depending on the goal, we pre-calculate different ZAR stores with a different chunking. And if the goal is just to show one gene, like gene X, we use this store where the data has been chunked per gene. And we just read the part for gene X and show it. But if the goal is to select a region of the sample and get the averages of all the genes, we cannot use this one. It would be too slow. So instead, we use this one. And we read in just the chunks for this region. And then quickly calculate the average and show it in the plot. So depending on what we want to do, we will use a different ZAR store. And this makes the database rather quick. So this is, again, a short demonstration So this is a ovarian cancer, and I will show the expression of this gene, EpCAM, by clicking here. And I can see it's relatively high here. So I think this is cancer tissue, but it's also present here a little bit. So I want to check there. And again, I can click with the mouse and select this region of interest. And for comparison, just select another region. And now I can compare between those two regions by clicking here. So now again, the average in each region is calculated and it's shown in the scatter plot. So each point is one gene. And so one of the genes with a high expression in set 1 is ESR1. So it's estrogen receptor gene. And if I go back here and I fill in ESR1 and I select it, so I can confirm that it has a high expression here, but not here. And this is showing the whole tissue slice, but of course I can zoom in, like click here, select a region, and then it zooms in there. And let's move it a little bit. So I can see the expression in every cell now, and I can see that it follows roughly the boundaries of this tissue. And also, of course, I can confirm that the expression is relatively low here. So, and this video was not edited, so I think I'm showing the real behavior of the database, and I think it's really fast, thanks to those ZAR stores. Okay, so because this is the end of the Xenium part, so because it has so many cells, we made a different interface compared to Visium, but it can do the same thing. So you can visualize the clustering, you can check the quality indicators, you can show gene expression patterns, you can see the metadata, so the spatially variable genes have been pre-calculated. You can just click them and they will appear here. And I can zoom in, I can select a region of interest, I can compare between them, I can see those scatter plots, I can confirm the gene expression of those genes with different expression levels. I can also show 2 or more genes in one plot using different colors. So I think it's really useful. Okay, I should finish my talk. So as a conclusion, first I want to say thank you to my team. So actually, it's a really small team. If you are interested in this kind of data analysis, please contact me anytime. And I should also thank my collaborators and MBDC and also AIMET for their financial support. So as a summary, so we made DeepSpaceDB. It's a spatial transcriptomics database, and it includes more than 2,000 Visium samples covering many tissues and diseases. And the interface, I think it's interactive and intuitive and useful for exploring the data. Okay, and this is my future plan, but actually we made the Xenium interface public, so you can access the data in our database now. And maybe in the future, we will consider adding other platforms also. And at the end, I should say, so this paper was accepted for publication in the database issue of Nucleic Acid Research. So if you search or if you click this link, you can see our paper. And also we wrote an introduction to the database in Jikken Igaku. So you can also, in Japanese, so you can read it there also. So thank you very much for your attention. That's the end of my talk.
Okay, thank you, Vandermoot Sensei. Thank you for your presentation. And although we are running out of the time, but we have several questions, especially addressing for you. So can you see it on them on the Q&A panel?
Yes, I can. Just a moment.
So could you answer them quickly?
So question 1 is, so it's about Moran's eye, shown in slides 15. So I think I can go there very quickly. So it's here, right? So the question is the Moran's eye used in slide number 15? Is it a global Moran's eye? I would like to analyze the data using other spatial statistics methods with geospatial data analysis too. So how can I obtain such information such as the coordinate data for Visium spots ID and the notation rule for the coordinates? Okay, this is not so easy to answer. So first, is this the global Moran's eye? I'm not so sure which version of Moran's eye this is. I'm just using the default one. I'm sorry, but I'm not so familiar with different kinds of Moran's eye. So as far as I understand, it's looking at all the spots, and for every gene it checks. So the autocorrelation. So I guess it's checking in each spot, the next spots or the neighboring spots, are their values similar? So in other words, are they correlated with each other? So, and in this case, here the spots with low values are not shown. But here there are many low value spots, but they are next to each other. And the values with high spots are also almost next to each other. So it has, I guess, a high autocorrelation. I think it's using the whole slide. Maybe that means global Moran's eye, but I'm not so sure. I'm sorry. And then the second question, so it's, I would like to analyze the data using other spatial statistical methods with geospatial data analysis tools. And the question is, so how can I obtain information such as the coordinate data of the spots? So this person wants to know how to get the coordinates. Actually, if you get the raw data of Visium, one of the files is, I forgot the name of the file, but it's something like the tissue location or tissue coordinates. It includes the X and the Y coordinates of every spot. And also, so this pattern, maybe I should show it on another slide. The pattern used in Visium, so it's a hexagonal pattern, right? The same file also includes the row and the column of every spot. So you can also use that to see what's the location of every spot. So in other words, you can see it in the raw data. And also, if you read your data into R, there is a function to extract the location of every spot. But what's the name of the function? I think it's something like a get coordinates or something like that. So you can also get it through Seurat, but I don't remember the name of the function. So that's the first question. Then I will click that one as answered live. Then I will go to the third question. So is the in-house, is it okay? So
yes, please.
Is the in-house data uploaded to DeepSpaceDB the data that was completed QC and other processes in Seurat? Okay, so let me go back to the end, maybe. So most of the data that is shown on the website is based on the quality processed data. But here in the download data, we added the Seurat object, processed, but also the raw one. So if you're interested in the raw data, you can also download it. If you're interested in already processed data, you can also download it. I think it should be easy to download them and read them into R and analyze them using the Seurat package. If you have any problems with doing that, please contact me anytime, and if there's some problem, I will try to fix it. So I think I answered that one. And then there's one in Japanese. Okay, so let me try to read it. So 空間トランスクリプトーム、解析用の PC はどの程度のスペックが想定されますか？あるいは、クラウド利用ですか？シングルセル RNA 作も、公開データで、3分になると、OK、128GB RAM 程度では厳しいのですが、so regarding the specs of the PC, if you are using Visium, actually Visium data is not so big. The number of spots is at most something like, it's typically 2,000 to 4,000 spots. So compared to the single cell data, it's quite small. And I think I can analyze it on just a normal laptop, actually. I can analyze it just on my normal desktop PC, which is not such a good PC, actually. However, if you're trying to use a Xenium data, I think you will need at least 64 gigabyte RAM, I think. I think 128 gigabyte RAM should be enough. But maybe for the really huge samples, I have not tried to analyze on my PC. So I'm running it on the server, which has 256 gigabyte. I hope that answers that question.